When you look at the devastating effects of climate change, well, few are as visceral as seeing glaciers retreat.
Hmm.
Across the planet, these huge masses of ice are vanishing, leaving behind these vast, raw, often called barren landscapes.
Yeah, absolutely. But that exposed land, that proglacial area, it doesn't stay barren for long, does it?
No, it kicks off this race, a frantic process of primary succession.
And that race, how life actually colonizes and transforms these brand new ice-free substrates, that's what we're diving into today.
Okay.
Our source material is a really foundational piece of global research. It's led by Gentile Francesco Ficetola, along with a huge international team. The paper is called The Development of Terrestrial Ecosystems Emerging After Glacier Retreat.
It feels like a really important topic, especially now because the pace of this change is just accelerating. So the question we're setting out to answer today is, what are the fundamental ecological rules, the ones that govern how life comes back in the shadow of this vanishing ice? Our mission for this deep dive is really to unpack the global data set they used in this paper. We want to understand not just what happens, but maybe the hierarchy of why it happens.
Okay, so let's ground ourselves first. Context. Glacier retreat is exposing these increasingly vast ice-free areas. We see it mostly in high mountains and high latitude zones.
Yeah.
It's one of the most visible signs of environmental change, globally speaking.
It's literally a blank slate, isn't it? Just
Yeah.
rock and glacial flour, this sediment that looks totally inert.
Exactly.
But then it rapidly starts to evolve into these incredibly complex biodiverse systems. Within decades, you can see alpine meadows and eventually, I guess, if the climate allows, even forests. It's like primary succession, but on fast forward.
Yeah, ecologists have watched this for ages, but the real puzzle has always been pinning down the primary driver behind this transformation. Historically, local studies, smaller scale, they propose three main mechanisms. Sometimes they even seem to compete.
Okay, let's frame those for everyone. Three competing ideas. The first is habitat filtering. I think of this as the real estate argument, Mega. Life only shows up if the local conditions, the soil quality, the nutrients, the microclimate right there are suitable. The habitat essentially filters who gets to stay.
That's one idea.
Yeah.
Then you have the biotic interaction hypothesis. This one says life doesn't just wait for the right spot, life helps create the right spot.
Ah, okay.
It emphasizes the relationships, like plants needing to establish first, which then supports certain soil microbes, which maybe then feeds insects or other animals. It's all about codependency.
Interconnectedness.
Right.
And the third one, that's the time or colonization hypothesis.
Yeah, and this one's perhaps the simplest. It's the waiting game idea.
Yeah.
It basically says, look, no matter how good the habitat gets, complex life just takes time to arrive. It's about dispersal limitation, the sheer probability of seeds or spores or insects physically reaching this newly exposed, often pretty remote patch of land. That probability increases the longer the land's been ice-free.
Right, just pure chance and distance.
Exactly. But the problem was, these local studies, they could really only focus on one, maybe two of these drivers at a time. We just lacked the standardized global data looking at everything together to really test all three against each other in different parts of the world.
And that's where this Ficetola paper comes in. It gives us that global framework to finally ask which of these is most important and, when.
Precisely. To solve a global puzzle, they had to go global.
Makes sense. They didn't just look at one or two glaciers. They took this massive snapshot of ecological history, right?
Oh yeah, colossal is the word. They built this broad-scale data set covering 1,251 plots spread across 46 chronosequences worldwide.
Okay, maybe unpack chronosequence quickly.
Sure. Think of it like an ecological timeline laid out in space. Imagine you're standing at the edge of a glacier today. If you walk away from it, every few steps you might cross ground that became ice-free 10 years ago, then 50 years ago, then 100 years ago, and so on.
So you're trading space for time, using different spots nearby to represent different ages since the ice left.
Exactly. And these sites covered all the major climate zones, tropical, temperate, subpolar. The ages were incredible, too, ranging from a plot just 1 year old, literally just exposed,
Wow.
all the way up to sites that had been ice-free for almost 5 centuries, 483 years.
That's a huge range. So how did they actually measure what was happening in these plots? What were they looking for?
They used some really cutting-edge methods. They measured 3 key things related to ecosystem formation. First, the standard stuff, soil properties.
Yeah.
pH, essential nutrients like nitrogen, carbon, phosphorus.
Basics.
The basics. Second, overall vegetation productivity, basically how much plant life is there. They estimated this using satellite data, things like NDVI, the Normalized Difference Vegetation Index.
NDVI? That's basically the measure of greenness, isn't it? Like how much photosynthesis is happening.
That's a great way to put it. Yeah, it gives you a sense of plant cover and activity. And the third, and maybe the most exciting part, was local scale biodiversity. They measured this using environmental DNA, or eDNA, metabarcoding.
Ah, eDNA. That feels like it's changing everything in ecology.
It really is, because think about the traditional methods, painstakingly counting plants, setting out traps for insects. It's incredibly time-consuming and maybe biased by what you can easily count or trap.
Yeah.
eDNA lets you just sample soil or water, find the DNA that organisms have shed, and then sequence it to see who's there. It allowed them to characterize the local diversity of 8 major groups simultaneously in a really standardized way across all these global sites.
8 groups. What did that include?
It covered the whole spectrum, really, from the tiny stuff, bacteria, fungi, protists.
The microbes.
Right, the microbes, all the way up to the macroorganisms, vascular plants, insects, springtails, earthworms, and even other animals. It gave them this amazing picture of the entire food web as it starts to build up from basically bare rock.
Okay, so they have this incredible data set, global scale, long time spans, detailed measurements of soil, productivity, and this huge range of life forms using eDNA. What did they actually find? What was the big discovery?
Well, the general pattern wasn't that surprising at first glance. Pretty much everything, soil nutrients, productivity, the richness of all these groups, it all increased the longer the site had been ice-free.
Okay, that makes sense. That's succession, right? Things get richer over time.
Exactly. But here's where it got really interesting. The speed and the pattern of that increase were dramatically different depending on the type of life form. They found this really clear 2-speed colonization process.
2 speeds. Tell me more.
Okay, so the early movers, the absolute pioneers, they're the microorganisms. Bacteria, and maybe to a slightly lesser extent fungi and protists, they were already remarkably diverse right away, like in plots less than 10 years old.
Wow. Okay, so the soil microbiome just throws a party almost immediately.
That's a great analogy. And it's an intense party for a while.
Yeah.
Their diversity shot up fastest in the first 50 to 100 years. They established this really robust microbial community super quickly, but then their diversity started showing signs of plateauing. Like it hit a saturation point relatively fast compared to the time scale we're looking at.
Okay, so microbes get in quick, build up fast, then level off. What about the bigger stuff? The plants, the insects?
The macroorganisms, plants, earthworms, springtails. They showed a very distinct lag. They were actually extremely scarce in those youngest plots where the microbes were already booming. Their local diversity increased really slowly for the first, say, 10 to 20 years after the ice retreated.
Wait a second. If the microbes are already there, and presumably they're doing their thing, breaking down minerals, maybe fixing nitrogen, enriching the soil, why the delay for the plants and animals? The habitat seems like it's actively improving, but the plants are just sort of dawdling for 2 decades.
That is the core question that comes out of this finding, and we'll definitely dig into the mechanism they propose. But crucially, what they saw was that once these macroorganisms finally get a proper foothold, maybe after 20 years or so, their diversity increase is sharp, it's positive, and importantly, it showed no sign of slowing down over the entire 483-year record they had. There was actually a positive quadratic term, meaning the rate of increase was itself increasing. They are still accelerating their colonization long after the microbes seem to have settled down.
Huh. So slow start, but once they get going, they really get going and just keep adding more diversity.
Exactly. And you can actually see this pattern mirrored in the environmental data, too. Soil nutrients, nitrogen, carbon, phosphorus, they increase significantly over time. And productivity, that NDVI greenness measure, it jumped from basically near zero in the youngest sites to much higher values in the oldest ones, tracking that surge in macroorganism diversity, especially the plants.
Okay, that makes sense. Plants arrive, things get greener.
Right. But there's also a climatic twist to this. They found these colonization dynamics were significantly faster overall in temperate regions compared to the colder subpolar or, interestingly, the tropical high-altitude sites.
Faster in temperate zones. Why would that be?
Well, the paper suggests the simplest explanation is likely temperature itself. Warmer conditions in temperate zones probably just accelerate everything else, the development of soil, the accumulation of nutrients, overall productivity. It gives life a bit of a head start,
Right, speeds up all the background processes that support life.
Yeah.
Okay, so this points towards a really interconnected system. It's not just one thing causing another. It sounds like lots of feedback loops. Is that where their modeling came in? You mentioned statistics.
Exactly. This is where they brought out the heavy statistical artillery structural equation models, or SEMs.
Okay.
Instead of just looking at correlations like X goes up when Y goes up, these models try to test causality. They build a map of how different factors might influence each other and see how well that map fits the actual data.
So what did the modeling show about the relationship between the non-living stuff, the abiotic factors like soil, and the living things, the biotic communities?
The model strongly supported what the researchers termed a coevolution model.
Coevolution.
Yeah, think of it less like a simple chain reaction, A causes B, which causes C, and more like this ongoing conversation, maybe a feedback loop, where different parts develop together. The abiotic bits like soil chemistry and the biotic communities, the plants and microbes, they develop together, constantly influencing each other back and forth.
That makes a lot more intuitive sense than just a linear sequence.
Right. And the data back this up. For instance, they showed that having a warmer local microclimate had a direct positive effect on soil nutrients. Specifically, it accelerated nitrogen accumulation.
Yeah, you can probably trace that back to biology, right? Warmer temps boosting nitrogen-fixing bacteria.
Precisely. It connects the climate factor to the activity of specific microbes, which then changes the soil chemistry, which then influences who else can live there. It's all linked.
So what did this mean for those original three big ideas? Habitat filtering, biotic interactions, and time. Did the models pick a winner?
Well, this is crucial. The model confirmed that you actually need all three. They're all essential pieces of the puzzle. The researchers tested this. They tried mathematically removing the influence of time, or removing habitat filtering, or removing biotic interactions from their model.
And what happened?
Every time they removed one, the model's ability to predict the actual patterns in the data just collapsed dramatically. It performed much worse.
So you can't explain the full picture without considering the habitat quality, the interactions between species, and the simple effect of time passing.
Exactly. It takes all three working together in concert to explain how these ecosystems develop over time.
Okay, but within that concert, was there like a lead instrument? Did any particular interaction stand out?
Oh, absolutely. There was a clear star player, an ecosystem engineer, if you will, the vascular plants.
The plants again.
Yes. They sat right at the center of this complex web of interactions the model revealed. They showed clear positive links with basically all the other groups they studied, the microbes, the animals, everything.
So once the plants arrive, they sort of structure everything else.
That seems to be the case. I mean, plants aren't just another organism. They provide the physical structure, the primary energy source through photosynthesis, shade, shelter. They change the whole environment locally. The study strongly suggests that once plants manage to establish, they become absolutely crucial for animal colonization, and they really drive that increase in overall ecosystem productivity we talked about.
Okay, plants are key. But this brings us back to that lag, right?
Yeah.
And maybe the most surprising finding you mentioned earlier.
Yes. This is really the kicker. Despite how important plants clearly are, and despite the fact that microbes are busy improving the habitat almost immediately, time since deglaciation remained the single strongest predictor of diversity for most organisms.
Stronger than habitat quality.
Yeah.
Stronger than the presence of plants.
For most groups, yes. The exceptions were those tiny, probably highly mobile bacteria and protists we talked about earlier. But for the bigger things, the plants themselves, the insects, the earthworms, time was king.
Wow. So what does that actually mean?
It's the critical takeaway, I think. It suggests the primary bottleneck, the main constraint for complex life getting established in these new landscapes, isn't necessarily the quality of the soil, because the microbes seem to get that sorted relatively quickly.
Right.
Fundamentally, it seems to be a logistical problem. It suggests that even when the ground is technically ready and the microbial community is buzzing, the larger life forms are mainly held back by dispersal limitation. They simply need the time for their propagules, their seeds, their spores, maybe adult insects blown by wind, to physically arrive at this new, often isolated terrain and successfully establish a population.
So it's less build it and they will come, and more build it and wait.
That's a great way to put it. Build it and wait for them to eventually find it.
And this whole picture, the coevolution, the importance of plants, but the ultimate dominance of time for macroorganisms, did this hold true everywhere they looked? Tropics, temperate, subpolar.
Remarkably, yes. That unifying conclusion that you need this joint role of time, habitat, and biotic interactions, It was consistent across all the geographical regions they studied, whether it was a high-altitude tropical glacier or one in the chilly subpolar zones.
That's powerful. It suggests we might be looking at some really fundamental, widespread ecological principles at play here.
It certainly does, and that consistency has some major practical implications, too. For scientists and managers trying to monitor these rapidly changing landscapes, well, it means you can't just rely on one type of data.
Right.
Efficient monitoring, understanding what's really going on, it demands a truly interdisciplinary approach. You have to integrate the ecological fieldwork, the genetic data from eDNA, environmental sensor data, climate data. You need all those pieces to get the full picture of how quickly these ecosystems are changing and where they might be headed.
Okay, that makes sense. Now, before we wrap up, we should probably touch on the limitations. Any study this ambitious must have some, right?
Oh, for sure. And the authors are upfront about them. First and foremost, as we discussed, is the reliance on chronosequences.
The space for time substitution.
Exactly. While it's essential for tackling questions at this scale, it does have inherent limitations. The big assumption is that space accurately represents time, that the older sites today truly reflect what the younger sites will become. But, the background climate itself might change over centuries, so it's not a perfect prediction.
Yeah, conditions aren't static. What else?
They also note that their sampling wasn't perfectly complete, especially in the really extreme environments like the high Arctic or Antarctic regions. So the data set has a bit more weight towards temperate zones.
Okay.
And third, there's the eDNA data itself. While it's revolutionary for telling you what species are present, the diversity.
It doesn't tell you how many there are.
Exactly. It fundamentally lacks information on abundance or biomass. You know a certain fungus is there, but you don't know if it's just a few spores or if it's forming a huge underground network, which you might get from traditional surveys. Linking that genetic presence-absence data to functional abundance or biomass is still a big challenge.
So bridging that gap is a key next step. Did they suggest other future research directions based on what they found?
Yeah, definitely. Building on the limitations, they stress the need to investigate the role of vertebrates.
Birds, mammals.
Right. They couldn't really capture those well with their soil eDNA approach. But vertebrates can have massive impacts. Think about birds dispersing seeds over long distances, or marmots digging burrows and totally changing the soil structure and nutrient cycling. These could be major players they miss.
Keystone species, yeah.
Exactly. And finally, they really emphasize the need to connect this powerful eDNA diversity data with actual field measurements of function. What are these emerging ecosystems actually doing?
Like, are they preventing erosion? Are they storing carbon, providing habitat?
Precisely. Evaluating those real-world ecosystem services is crucial for understanding the value and trajectory of these rapidly changing landscapes.
Okay. So to sort of bring it all together, the big takeaway for me seems to be that life's return to these brand new landscapes, these proglacial areas, it's this really intricate, co-evolving dance, timed almost perfectly. You have the microorganisms, the invisible pioneers, rushing in, fixing the soil with incredible speed, but hitting their diversity limit relatively fast.
Right.
But for the complex visible life, the plants, the insects, the worms, the story is more about patience. Even when the stage is set, when the soil seems ready, they're fundamentally held back by the sheer time it takes to physically get there.
That sums it up beautifully. It's a story of microbial speed versus macrobial patience, all orchestrated by time and opportunity.
Yeah.
And this, I think, leaves us with a really important, maybe slightly unsettling question for you, the listener, to ponder, especially as we see glaciers retreating faster and faster. Given that time since deglaciation is the strongest predictor for complex life finally showing up, how will the increasing speed of modern glacier retreat, which is way faster than many historical retreats these chronosequences represent, how will that affect the long-term diversity and structure of these unique emerging ecosystems? Do these brand new terrains even have enough time now for the seeds and the creatures to arrive and establish before the climate shifts again, or before the next disturbance comes along? Are we creating landscapes faster than complex life can colonize them?